thanks everybody for coming out today. I know you probably got a million emails from myself and Jeff and various other people, and we really appreciate everybody being here in the room with us today to talk about Dotmatics Flow Cytometry Solutions, and in particular enabling scientists from basic research to personalized medicine. So I got myself here, Sean Burke from De Novo Software. I'm joined on the stage today by Prier from OMIQ, Ryan Brinkman from Dotmatics, and Garrett Collett from EMS. We're really excited to have Garrett here talking with us today. He's going to discuss applications for spectral flow cytometry and CAR-T characterization. If you haven't done CAR-T work or you don't know much about it, this is a great place to learn. And if you have before, well, this is a great place to learn a little bit more. Now, I think last year was the first time we talked about Dotmatics at Cyto, or maybe 2 Cytos ago it was. But for those of you who aren't familiar, what we're trying to do at Dotmatics is build the world's most powerful scientific R&D platform. And we do that by connecting best-in-breed kind of applications and software that enable collaboration and automation and analysis. So, what that kind of means, and one of the problems that, I'm sure has been talked about over and over again at Cyto this year and probably for many, many years, is that a lot of people state workarounds as being required to get data into meaningful outputs, right? And that negatively impacts their work. And when we talk about personalized medicine and CAR-T analysis, CAR-T therapies, that is patience, it's not just work, right? A large proportion of people report compromised decision-making, due to informatics challenges. And again, when that comes back to personalized medicine, that's people, right? It's people that are getting compromised decision-making. We want to avoid that. So, we look at this kind of theme at Dotmatics of, never compromising, using the software you love. So hopefully there's software that you love up here, and if you haven't used any of these yet, hopefully you'll come to love it very, very soon. Dotmatics, OMIQ, FCS Express, Prism, Snapgene, Genius, some new players in the game here with us, MSTAR and Softgenics, are all part of the Dotmatics platform. So we're trying to offer solutions, not workarounds, right? Solutions and workflows, not workarounds. And kind of in the middle of this hub here, we see Dotmatics Luma. We're excited to be talking about that today, and Julia's going to talk about it a little bit, Ryan's going to talk about it a little bit, and I encourage you to come back down to the booth to talk about what Luma is a little bit more. But when we talk about flow cytometry and personalized medicine, CAR-T cell therapy is this huge breakthrough in medicine, and it gets me really excited when I think about it and I talk about it, and especially when I go back and I speak to the development team, but trying to explain what CAR-T therapy is and how life-changing this is, and how people are actually having cancers cured, right, by this kind of technology. And flow cytometry is being used at many different stages in this process, and Garrett's going to give us a really great overview of where and why and how. But in cell preparation, production, QC, and it's not just limited to cancer treatments, right? It's really limited by scientists' imagination at this point. What you can do with CRISPR, what you can put into these cells. And because the cells are going back into patients, validation and regulatory requirements have become more important. Even as recently as January, FDA issued new guidance for CAR-T cell therapies specifically related to flow cytometry. And one of the other challenges is scalability and cost and geography, because these treatments need to be close to the patient. You can't just take cells out and send them across the ocean back to New Jersey and then bring them back and put them in the patient. I think I ran into somebody in the hallway, we were talking about that just on the way up here as well. But really, what everybody's doing in this room, all the people who are involved at Cyto, people who are involved in flow cytometry, everything that you're doing is important, right? Whether you know it or not, if you're working on CAR-T therapy or not, if you use a reagent, you optimize that in some sort of way, you're using an instrument, you've done something with it, and that technology eventually, that knowledge goes into personalized medicine. Those contributions are super important, and everybody in this room is really making a difference with personalized medicine. And I appreciate everybody for everything that they're doing in this room and flow cytometry in general. So I'd be remiss if I didn't talk about software that I love. software that I love is FCS Express. I love OMIQ too. Right? I've come to love OMIQ more over the last few years. Right? And we laugh, but I really do. I really like OMIQ. It's got some really great features and functionality. But with that being said, I'm going to leave that to Julia. I'm going to talk about FCS Express for a little bit. And if you don't know about FCS Express and personalized medicine, we're being used by many of the leading CROs and CDMOs, many of the major players in the game that are manufacturing and have approved cell therapies. None of them will let us put their name up here, so it's one of those limitations, but trust me, they're out there. But of course, it's being used by people for research all over the world as well. But the reason that these labs like to use us for validated and personalized medicine is because FCS Express scales from R&D through RUO versions, research use only, in vitro diagnostics, with and without what's called 21 Part 11 ready add-ons. Don't be too scared if I use terms like 21 Part 11. We can explain what all that is at some point. But what we did is we put out a new validation ready package just within the last few months. To make it easier and scalable for folks to get into this kind of validation process. Because it's something you need to be thinking about way ahead of time, right? So if you've done work in a few different ways, and then you kind of come to, I need to get this into a patient now, and then the QC people go back and say, okay, well, where's all the regulatory work that you did leading up to this, or what software we're using? And then they might say, hey, you got to come back and do it all over again, or revalidate it, or do something else. So it's something to keep in mind if you're going to be working on any type of validated research that kind of plan out in the future. Anybody who's seen me talk has probably seen this slide for a long time. I should probably update it. But the reason we use FCS Express more generally is technology convergence, right? So that cell phone that's up there, that was the first cell phone I actually ever had, so you can do the math on how old I am. GPSs, cameras, calculators, all that stuff wrapped into a smartphone now. So life is easy. And we've seen that same need for convergence with flow cytometry, bringing tools from Prism and Excel and PowerPoint into FCS Express. And now we have that kind of bi-directional linking there with Prism. I should change my arrows so the Prism people don't get upset. But what the difference is other softwares generally produce basic plots and basic statistics. And with FCS Express and even tools like OMIQ, it's fully integrated. The analysis, the statistics, the graphing and reporting. So you can get done in one software in one place and get a result out at the end. That's also why validated labs love us, because they don't have to validate many different pieces of software. Now, since Cyto last year, I've got grayer, I've got wider, but we've also made many improvements to the software. We have the GraphPad Prism integration. We've enabled multi-instance in FCS Express, so if you want to kick off a few TISNEs and something else in the software, and then continue analysis in FCS Express, or do something else, you can do that. We've upgraded our quadrant interface, so quadrants are now automatically created as gates in the software. And there's a huge number of user interface improvements in there. So if you ever found something challenging or frustrating in the past, we put a lot of effort in the last year to make that much better for you. Now, I talked about that validation ready package, and really it's designed for researchers in the cell and gene therapy space, folks working in regulatory environments, or really any lab needing any sort of GXP compliancy. And essentially it comes out of the box. It's a self-hosted network, so that means QC and IT people love this because they put it on their own network in the lab. All sorts of definable security groups and permissions, and e-signatures and audit trails. So I know folks doing day-to-day research, they don't really care about any of this stuff, right? But again, you need to be thinking ahead for the future. And the reasons you consider a validation package from FCS Express is one of the reasons is it integrates into new and existing workflows. It makes the software very reliable and repeatable in everything that you do. And we have the ability to add on IQ, OQ to that. It means installation qualification, operational qualification, and connections to LIMS and LIS systems. But I mentioned the scalability. And I think the scalability is really cool here because if you're working in University A, and you develop some sort of FCS Express layout for CAR-T analysis, and then you kind of finalize that panel. What FCS Express allows you to do is move that into an RUO version with our validation-ready features enabled, right? The same layout, right? You don't have to recreate anything from scratch, and then you're working in this kind of validated environment. Then when you're ready to take that and move it into a patient, because having those locations close to the patient is important, you can move that into our IVD version, right? And then you lock it down, and you send that template to all the different labs that are manufacturing these cells throughout the world. And that's what these labs actually do. These are not real locations, because probably people at BMS would be mad at me if I put up real locations, because Garrett's here. But other labs are actually scaling like this, and it makes it very easy and very efficient for folks to work with. Now, I put Luma down here. What is Luma? We'll talk about that. But all that data has to go somewhere, right? And generally, folks use LAS and LIM systems to do that. Dotmatics Luma might be a place that we'll be able to send some of this information to centralize it as well. And I'm hoping I'm getting everybody excited about Dotmatics Luma in some sort of way, but you got to come down and ask about it. But I wanted to take a look at the Prism integration. So this is FCS Express, the latest release, where we're going to export directly to Prism. And it's in the batch actions, right? So you actually just create a Prism project in the software. You tell where to save it, and then you pick what sort of table you want to put your data into. And then you use some of the annotations from the data file to kind of group things, right? And like everything else in FCS Express, you drag and drop to get statistics into that Prism export. And when you click Run, it's exporting all the information, it's batch processing it all to a Prism file that we open up, and then we can insert a graph in Prism from our data, right? So there was no copy and paste, there was no kind of, in-betweens, there was no workarounds, right? This was a direct solution to go into that. And you're going to see some more of that from OMIQ too, which is really, really cool. And so I see some people smiling like, oh yeah, this is the best thing ever. I like to see that. Now, you come down to the booth to talk a little bit more about what's coming from FCS Express. But again, moving on to talking about software that you love and that I'm coming to love, OMIQ, and I'd like to introduce Julia here. Come up and talk a little bit about OMIQ and what's new there. Thanks, Julia.
And thank you all for being here to listen about the exciting things that are happening with Dotmatics and with OMIQ. So I'm Julia Prier, I'm the new product manager for OMIQ. Until very recently, I was the product manager for Genius Prime, which is another software with Dotmatics. And in a past life, I was an academic, so my PhD and postdoc was all in immunology, which is to say that I spent all of my 20s in some Spectral flow cytometer, as you're probably familiar with. So come and talk to me about how OMIQ can help you with your flow analysis. I'll be at the booth and keen to hear how we can help. So for those of you that don't know what OMIQ is, it's a cloud platform for classical and advanced flow cytometry analysis. And there's three aspects of it that make it a particularly powerful platform. So it's cloud-based, which means you've got cloud storage and computing, so you don't have to worry about your laptop going off fire when you're trying to run advanced analysis. You can also share data and workflows easily with your colleagues, and there's seamless integration and automation. And we really think that in OMIQ is where flow cytometry meets data science. So there's two principles of data science that we've used in OMIQ. The first is metadata, and we heavily encourage you to make use of this feature because it really allows you to capitalize on the ability to view, so you can virtually concatenate your data, calculate and streamline your analysis, both in OMIQ and Prism and beyond. So I'll demonstrate that later. The other principle is workflows. So here you can see workflows. So it's a visual cue. You can see everything the data's been through very explicitly. And this, you can also have templates which allow you to version this and audit it, and most importantly, have reproducible analysis. Okay, so into the nuts and bolts of what OMIQ does. It's got plenty of tools for both classical analysis and advanced analysis. So everything you would expect for classical analysis with scaling, compensation, gating, of course, subsetting, figures, export stats. But where OMIQ really shines is in the advanced analysis because it is cloud-based, so you can kind of set and forget. It's got tools for dimensionality reduction, all the different flavors of TISNI that you're used to, UMAP, PCA, Lots of tools for clustering, for example, FlowSym and the rest, as well as trajectory and pseudotime analysis, stats and differential expression analysis, and data cleaning and normalization. And for those of you that have learned R and Python, you can also have custom analysis to run those within the same workflows. Yeah, so not only do we have a lot of tools, we're constantly adding new tools to this, and improving the existing tools that we have. And here I've got highlighted a few of those that I'll be talking about today. So I want to talk about them in the context of the current focus of OMIQ, which is this end-to-end workflow, and really trying to add a lot of efficiency to that. So first I'm going to show kind of like a worst case scenario. Take from it what you will. You'll probably all be, different kind of experiences. And then I'll show what it could be with OMIQ and other Dotmatics products. Yeah, so first with any workflow or experiment, idea, project, what have you, it starts with an idea. You then have to prepare your experiment, so you might be gathering information about your reagents, your samples, your SOPs, often from dispersed locations. So now you've already got data silos and a lack of connectivity there. You then perform your experiment. I can't help you there. I don't know how fast you prepare, so this could be variable. And after you've performed your experiment, you have to move that data. So probably people aren't using USBs anymore. No shade, guilty of that in the past. Or you might have a network with your IT set up. But either way, you're moving that data, there's manual data movement, which often results in duplication as well. You then perform your analysis, so you might have to manage plugins and their dependencies. It can be difficult to track the analysis, and you can have limited computing power. And then sharing those results with your colleagues and collaborators. Often there's antiquated sharing methods, which again result in more duplication of those results. So, with OMIQ, again you start with an idea, but using other Dotmatics products, such as the electronic lab notebooks, laboratory information systems, and bioregisters, you've suddenly got all this information that is now connected, and all that metadata is together. You then perform your experiment as per usual, but we've now got this new tool that I'll be talking about called Luma Lab Connect, and this is a tool that means that you can send the data directly from your machine into OMIQ. It's also a fleet management system for any Collett managers here. You then perform the analysis and the sharing step in OMIQ. So as I mentioned before, there's a bunch of benefits to this, so natively integrated algorithms, easily track the analysis, cloud computing, easily collaborate, and integrate with other software, which ultimately leaves significantly more time, less effort, so you can spend time on the science, which is what everyone wants to be doing. So now we'll talk briefly about these three kind of new features here. So first is Luma Lab Connect. So as I said, you can pull your files directly into OMIQ from flow cytometers. This can also be automated. Here you can see it's a data set, and you've just got the button. Yeah, pull it in. And I'm just going to deviate for a second here and show a demo for auto-gating, because suddenly you can picture the situation where you can automatically pull your data in and have a workflow set up with auto-gating. So then the next step, when you get into the computer, is to just look at the results instead of doing any of the previous steps. So here you can see, click this, the custom auto-gating. So you can see a few nonsense gates here. And if you click auto-gate, voila, everything looks great. So yeah, this is auto-gating, can be customized to your own data. It's available natively in OMIQ, and it has editable gates. So at the review step, you can go and make changes. All right, so you can also send data back to Luma. Keep this in mind because it'll make sense at the very end. But here is an example of the data in Luma where you've got your batch results, the metadata, this data file associated with it, and your stats that you're showing there. So bear in mind why we want to do this. I'll come back to it at the very end. All right, so another thing we've been working on very recently, and it'll be released very soon, is a Prism integration. So you can generate Prism files in OMIQ, as I'm showing here, through export stats. This is just a sample. So here is Prism as well as CSV options. And then it will generate a Prism file that you can download. So what I'll show in the next video, after this one, is how to rearrange that data in Prism to completely avoid the whole copy pasting in a spreadsheet editor step. Okay, so one downloads the file, opens it up in Prism. This is going to open up in a multi-variable table format. Here I'm just changing one of the data types. Go into Analyze, Extract and Rearrange, and from here you can just tell Prism, for example, I want to look over time at my CDA T cells, group it by the sample type. This is metadata that was in OMIQ, and the replicates are by patient, more metadata that was in OMIQ. This is going to do all the reshaping in Prism, and I get my result. So from here you can go on to do your stats and what have you. So before I move on to the next slide, I want to mention this is a very active area of development. We're very keen to work with Prism, and we are working with Prism to add more efficiencies here. So to batch those response variables, to add stats as that step to automate it. So if this is of interest to anyone, please come and talk to me. I want to hear all about it. All right, and more. So with OMIQ, there's a whole bunch of other things going on. So we're working on turnkey validation. We already have clients using GXP servers where we're trying to make that even easier. We're finally getting the 3.2 file import. Graphular virtual concatenation is coming. More algorithms for normalization and differential analysis. Improved reporting with the freeform layout, enhanced box plot options, stacked and group bar graphs, and changes to gating. So all of these things we can demo at the don't have time today. And so finally, I want to close on mentioning Luma. So as I said earlier, you can also send that data from Luma Lab Connect back into, from OMIQ back into Luma Lab Connect. And what that does is it'll send it to Luma. So with Dotmatics Luma, this integrates with all of the other analysis software, so you can have sequencing analysis, and all of a sudden you can perform multi-omics. So this is a really powerful platform. It also is set up so you can capitalize on future AI tools that will be developed here. So ultimately what happens is you're just supercharging the idea generation here. And so, yeah, again, it's more time for you to focus on the science. And as far as the future OMIQ direction, we're customer-led. We want to know what you want to do, so yeah, come and have a chat and see how we can move the field forward together. So thank you for your attention.
I guess I'd usually thank the organizers, but now it's a pitch. I'm not trying to sell you something. I am actually trying to sell you something. But I'm trying to sell you an idea. That's actually a bit more than an idea. So it's been an idea before, but now we have an implementation of the idea. And the idea I'm trying to sell you on is something that is not a Dotmatics product. It's an idea that Dotmatics is, we're all about integrations, and we also want to help the community. And what I'm going to talk to you about today is not going to be something that we're trying to sell. It's something we want to open up to the whole community, this idea, this representation. The problem that we want to solve for everybody, the problem I want to solve for everybody, and through Dotmatics and through collaboration with everybody here in the audience, and also the other pitch I have is there's a workshop later on today that we're going to talk about this, hopefully more with the community, about annotation. And the problem that I'm trying to solve is annotation sucks. It sucks for lots of reasons, especially in the context of fair data. And the challenge that I have is you all use words to describe things like CD positive or this is a T cell. But the person next to you uses different words for the same thing. And this is a challenge for me because I want to help Big Pharma and other people analyze large data sets. And the challenge is when you're using different terms to talk about the same thing, it becomes a real challenge to integrate data sets across centers, across studies. And that's a problem for me because I'm really interested in doing big data science. And so I want to solve that problem. And there's also the challenge when you have different types of ways to do analysis. We have different dimensionality reduction algorithms, and there's going to be more of those. The problem's only going to get worse because you can find the same thing in different ways, and all these different algorithms have different ways to say this is what we found. So I want to solve that problem. And the way I want to solve that problem is to get everybody to agree what a T cell is in a binary applied. Maybe that's easy. It gets more difficult as we go down, but I think now is the time we can do that, and I think that's going to help everybody, so that we can use the same words to describe the same things. That's going to enable big data science. The idea that we have is to have an atlas of populations that says, as this bivariate plot and this base of the hierarchy, these are the populations that you're going to find, and these are the labels that we want to put on them. Personally, I don't care what you call it. You can call it a T cell or a B cell. That might be important to you, and it's probably important to science. The idea in the background is to be a computational label that software can use to interchange and use science on that. But it's also going to, I think, help the field as a whole if we can all start using the same words for the same things. That's just, I think that just kind of makes sense. And so the only hypothesis that has to be true for this to work, as far as I know, is that every time I see a bivariate plot, it's scaled to the interesting data range. The white space doesn't really matter. And if this is true, and the proof of concept I have seems to support the idea that everything I'm going to say after this is kind of going to work. So the whole idea is, if we, this is some examples of how this would work. So the first thing what we have to do is we have to build this master map that's shown here, that if you have an unlabeled bivariate plot, we're going to compare that to the master map, and then we're going to assign the labels based on that. And that master map would be owned by ISAC, IUIS, ESCA. A whole bunch of experts in this room would agree that this is a T cell. This is going to happen over time. And I realize there's some things down in hierarchies we're never going to agree on what the labels are, but if we get 90% of agreement on 90% of the populations, I'll be really happy. In order to build this master map, the idea is to get all the data in the world, good data from good labs, and maybe from Flow Repository would be a good place to start. That's all published review data. But experts in the field here or in this room who have experts on T cells, I love your data. So I'm really into this big data idea. That's going to come up in a second. So get all the CD3, CD4 plots in the world and smush them down into one. So that this is all the kinds of things you would see in this plot. So how can we do that? We take those individual data files, zoom into the SR data range, so get rid of about 2.2% of the data around the outside, convert that into a pixel map, and then take the Z-score, which is the heat, how high how many events are in that area, and kind of squish it down a bit, so that the rare events all show up. And do that, we show here 2 different examples of how we can process that data. And then what we can do is we can take all different data sets. So here's an example of 3 different data sets that we pulled out of Flow Repository, had CD3, CD4 in them. Within that data set, we can take all the data from all the different plots within one data set, and then combine them all within one study, and then take all the different studies, and then combine them all again. And that shows here at the bottom. So here we took a few hundred files of CD3, CD4, and that shows the variability that you have at that step in the gating hierarchy based on the heterogeneity. So these are all the things that you would ever expect to see. And then what we have to do is we have to draw boxes around them for what the populations are. Well, I have a really good citizen science infrastructure that can do that. I have gamers, and I've talked about this before, I have hundreds of thousands of gamers who are happy to gate flow cytometry data for me. It kind of works. We put OMIQ, or FCS Express, if you will, inside the game. These are the gates that the expert gamers can do. So these are, really, believe me, these are 2 random plots that pull here on the left. I think it looks pretty good. What the idea is, what is everything that you would expect that you would ever want to put a label around? They are putting a gate around that. And even better than that, I have about 500 gamers look at each plot. The next 2 columns here on the right, we had 4 different people give us what we consider a good result. It took us about a year to figure out how to find the good people among all the bad ones. Then we can combine all those to get a consensus. So here's the first start of the idea of what the map would look like at each step of the gating hierarchy. And I can now go through and generate all these maps down here. But what we don't get, and the problem I'm trying to solve, is if you have an ML algorithm or some other algorithm, is the labels on these. So the idea for the computational label is we just align one of those populations on a grid, 64 by 64. We can identify computationally what that population is by the pixels it overlaps. And then what we can construct is a pixel-based ID for the locations that these populations are as you go down that hierarchy. And then what we can do is we can put a bunch of people in the room and say, well, this population down here on the bottom left at the end is called a DNB cell. And that would be the official label that the atlas would have, that some experts would agree on, that we could then assign to any population. But in the background is also the computational label. And I think one of the things that will work is this could be a lingua franca, or a translational method that different dimensionality reductions could use this bivariate gating approach to give the same label independent of what algorithm you used. So we can all, again, be sure we're talking about the same thing, because if you use like a flow sum, you can probably identify that some events through a bivariate gating profile that match the events that you found that are interesting in flow sum, and you can use this ID to translate between different methods. And I think that's going to have a lot of advantages to make sure we're talking about the same thing. And then how it works doesn't really matter. It could be done as a web service, or ISAC would just, here's the data, you could download that. You know, if you're using the FCS Express, it would sit on your local computer. And then when you have an ungated file, you push a button, some information gets translated back to this, compared to the atlas. We align how many pixels overlap with the master map. Then we can assign both the composition label and what the ASTAC or ESCA or IUS approved name for this. It's a T cell, it's a D cell, it's an NK cell. We could also tie that to ontologies. That would give us a lot of extra information about populations and give you really cool science with that. And then we feed back to your ungated file, and you push a button, and this is the label. And I think that would be great. And everybody will get that same label. And this is exciting to me because this really will enable big data science. And so all the pieces I'm showing here now have implementations. We can do different parts of this. So you've heard about Luma Lab Connect, so we can ingest all your data off the machine today. This is working in Big Pharma today. We have ML gating that's in beta with us, and other people have auto gating tools. Then we can label all your populations automatically. Then we can do statistics on all of those. You know, we have Prism, there's other statistical tools as well. We know the kinds of statistics you want to do. That can be automated. We can layer that on top as part of the pipeline. After gating, I want to make these kind of plots. And then the other thing we can do for you is say, these are the interesting plots. Here's a p-value that shows this plot is very interesting. And then you come in at the end of the day, and before you leave at the end of the night, you push the button. Next morning you come in, it's been read in, it's been gated, it's been labeled, we found the interesting plots, and then you can do the science. And you don't have to do all that other work that may not be as interesting. And that's exciting to me because now we can do that, not only the data that you're collecting today, we can go back and reanalyze, relabel all the data you've ever collected, past and future. We can put that, for us, we would put that in a harmonized, standardized data platform called Luma. And that is going to really change, I think, it's going to change the fundamental value of all that ROI data, and it's going to enable better science. And again, this is something, an open project. If you're interested, please talk to me. Come to the workshop this afternoon. We'd be really happy to talk to you. Thank you very much.
Thanks, Ryan. And thanks all of you for coming to this talk today. Lots of lovely faces in the room. My name is Garrett. I'm a scientist at Bristol Myers Squibb at the Seattle, Washington site. And I'm really excited today to be talking to you about applications for spectral cytometry in CAR-T characterization. And the reason this talk is relevant in this context is we use a lot of the tools that were described today to do our spectral flow characterization. So just a quick agenda. I'm going to start the talk by giving everyone just a quick background onto what cell therapy is, with a little overview on analytical development strategy. But I'll leave the bulk of the talk to talk about real applications, development highlights, which include automated titrations analysis, flex channel strategy, and infinity flow. And then I'll wrap it up with some concluding remarks. Okay, so let's dive into a quick background. I imagine most of the room is probably pretty familiar with cell therapy, but in case you're not, I'll just do a quick walkthrough. The process starts with leukapheresis, in which a white blood cell rich fraction is removed from the patient or donor. That material then undergoes a selection to get out the subset of interest from the crude leukapheresis and is then activated. And activation is followed by gene transfer, where the CAR construct or other construct of interest is introduced into the T cell. And then following that, the T cells are then grown up in your favorite media so that they can make dose and then be infused into the patient. So that's what CAR-T therapy is in a nutshell. And you know, that's kind of a simple story, but this whole process is a little bit more complex. This whole process ranges from, you know, in the overall drug development strategy, ranges from ideation or proof of concept for maybe you have a new drug or a new construct or a new manufacturing process that you're try out. And your process moves from ideation to drug candidate endorsement by whoever your leadership team is, and then on through to process analytical development, which is where I live in the organization. Finally, the process is transferred to the manufacturing setting, and then if all goes well, you then have a commercial product. And there's kind of two main bins that I'm trying to bin the software that I'm about to talk about into kind of two major bins. One bin I'm calling discovery, and one bin I'm calling compliance. And at different phases of the drug development process, you want an emphasis on one or the other. Just generally, you want to do more discovery and less compliance early in the process, and more compliance and less discovery later in the manufacturing process. And those bins really nicely correspond with the two softwares that we're talking about today, which are OMIQ and FCS Express. I have to admit I'm a little more familiar with OMIQ than FCS Express, but I'll just give you a quick rundown of how BMS uses these two softwares. So OMIQ is a really powerful solution that integrates both your traditional manual analysis with high-dimensional exploration. The cloud hosting aspect of it allows computations to run really quickly and really enables easy collaboration. No more sharing copies of your FlowJo workspaces. You can directly share your workflow and your analysis with your colleagues. OMIQ also hosts a variety of algorithms that are of interest to the cytometrist, like automated cleaning or clustering or analysis algorithms. And you don't need to know R to use them, which is really nice. FCS Express, I've generally been into more of the compliance category. BMS uses this mostly in the manufacturing setting, although by no means are OMIQ and FCS Express only usable in either end of this spectrum that I've made up. So FCS Express has research use only and also clinical editions that allow scaling for a variety of different regulatory needs. And the 21 Part 11 compliance allows further scaling for various site regulation requirements. And the templating aspect of FCS Express also helps reduce time between execution of your assay to real data and real decision making. So these tools have directly enabled the data that I'm about to talk about. So that's maybe a little plug for the Dotmatics folks. Okay, moving into CAR T biology. This isn't a T-cell biology talk, but I'd like to quickly run through why CAR Ts are so complex and so hard to characterize. There's a whole bunch of different methods of action that a CAR T can do to kill a tumor. CAR Ts can lyse directly via perforin or granzyme, or through the Fas ligand axis. But not only can they kill directly, but they can contribute to the broader inflammatory milieu, like releasing inflammatory cytokines. They can recruit other cellular mediators of inflammation and move around to the site of tumor. And then they can also change how the individual CAR T functions do. They can proliferate or differentiate into different subsets with different functions. So this is all a lot to measure and a lot to understand out of your product. And we try our best to learn as much as we can about such a complex drug. And this process is really broadly called characterization. So I'll give you an example of a kind of a first generation characterization strategy. Say you're a researcher in your CAR T group, and you might think that memory phenotype has some sort of association with CAR T function. You might develop a conventional cytometry panel with a handful of markers that you know are associated with memory phenotype. And then you might, as your clinical drug product comes in, you might characterize the drug product using this memory panel. And at the conclusion of the clinical trial, you can crunch all the numbers and see if any of the memory phenotypes that you've measured have any association with either manufacturing or clinical outcomes. And this approach is great. There's nothing wrong with this approach. It's been really successful so far in the CAR T space. But I think maybe there's room to change this from a linear process to kind of a more cyclical and more next-gen way. So I'll give you an example of a second-gen characterization strategy that myself and my group think a lot about. So now kind of we're expanding from just kind of a singular aspect of T-cell biology to several different modules of T-cell biology, like activation and exhaustion in addition to memory. We're developing not just a single panel, but multiple large spectral panels with 20 to 30 markers that encompass the above modules. And we're deploying these assays on pre-selected cohorts of drug product. And the goal of this, is to deepen our product understanding, elucidate method of action, and inform product and process development so we can make a better drug. And in tandem to this, we're thinking about a lot of other things as well, like standardization of panel design and analysis strategies so we can multiply our learnings across assets. We're thinking about flex strategies too, and this is a concept that I'll bring back in a little bit, but these flex strategies allow us to test new markers on the fly. We're using high-dimensional analysis to identify new subsets of interest and also to guide our manual or low-dimensional gating strategy. In parallel to our spectral flow, we're utilizing functional assays as well so that we can correlate phenotype with function. And finally, we're thinking about new models like infinity flow, which I'll come back to later in the talk, as well as chronic stimulation and scale-down manufacturing models to interrogate these attributes or characteristics at different states or time points throughout the manufacturing process. And the goal is, with each one of these kind of novel ways to approach CAR-T characterization, we can learn something new. So I hope I've convinced you that we think a lot about characterization at BMS. And we'll just dive into the data and dive into real-world applications. So I'll talk briefly about spectral cytometry at BMS. We have a lot of different spectral panels that we're using on the regular. It's not enough time to go over all of them, so I'm going to focus on just 3 for this talk. These 3 panels we're calling T-scope, which is a T-cell immunophenotyping panel. All-PBMC, which is a more broad immunophenotyping panel for like the entire milieu of the starting material. And I'll also talk about infinity flow, which is kind of a special application of cytometry. So let's start with all-PBMC. As I said, all-PBMC is kind of a broad phenotyping assay, and the reason we developed this is that BMS has had a lot of success in the hem-onc world, but we're moving towards autoimmune indications. And as we know, starting material has a pretty big impact on the ending drug product. So we want to understand how different starting material compositions might influence drug product manufacturing. To develop this panel, we leveraged a whole bunch of resources that were available to us. Subject matter experts that are in-house, as well as the OMIQs, and a really great paper from Alex Hubik at the Allen Institute for Immunology that I would strongly recommend everyone checking out if they're interested. And this final panel kind of incorporated all of these, the OMIPs, and then kind of our existing in-house panels, as well as other markers that we anticipate to be related to drug product quality. So this is what all-PBMC looks like. I won't go through all of it now, but the purpose of this slide is just to appreciate the depth and breadth of this panel. It's 34 colors, so it's fairly large. I wish I could say what all of the markers were, but legal has forbidden me from saying this one. Okay, so all-PBMC 34 color panel, it's big, and anytime you work with a panel that big, one of the biggest challenges that we come into is titrating all of our antibodies. So I don't know who all went to Anna's talk yesterday, but I hope I don't have to tell this group that titrations are really important for cytometry assays. If you titrate your reagents, you reap a whole bunch of benefits, like retaining the correct staining profile. You can see from the example that I'm showing here, using too much or too little antibody can really obscure the true pattern of antigen. You can minimize spillover spread error by titrating your antibodies properly. You get better lot control from vial to vial and lot to lot, and oftentimes you don't need as much antibody as the manufacturer recommends, so your reagents can last longer. And lots of different ways to interpret titration curves. I've shown a few common metrics that we use here. So titrating all of these is a huge pain. So what we ended up doing is developing an in-house titration automation tool. This hasn't been shared yet. Again, BMS is a little stingy on sharing tools. But essentially what this tool allows you to do is to acquire all of your data and then plug it into this fancy little tool. And what you get out of it is all of these different curves and different metrics that can aid you in picking the correct titer for your antibody. And this is what just an example of what one of the readouts look like. We get these nice little graphs that show stain to noise, signal or stain index, and things like that. So, been very useful for us. If you're a PI or somebody that is interested in data science, I would strongly recommend supporting your lab members and learning how to code so that you can make these fun tools for you and your group. The kind of crux of this is that we have the tools to create really big panels now. Panels between 30 and 50 colors are more or less commonplace. But no matter how big your panel is, it is impossible to predict what all of the markers that your customers might be interested in, or you yourself might be interested in. You could add or remove these markers ad hoc, which works fine, but it really hinders your ability to speak longitudinally to your data and to compare to past and future data. And you could develop new panels to accommodate these kind of custom requests, but that's really time consuming and really inefficient. So what do you do instead? you designate specific channels in your panel as flex channels, where you can swap different markers out on the same floor for. And this strategy is not new. This strategy has been previously described for a long time. We did not invent this, but I'm showing here a nice little application of it. And as you do this, it's important to choose colors that have minimal overlap to the rest of your panel. You want your panel to be robust enough to use a variety of markers at a variety of different antigen expressions and still retain the quality of your data from the original panel. So one of our goals here is to characterize the impact of using different markers in our panel. So I'll switch again and talk about this T-scope panel. This is kind of the first time we really encountered this problem, is developing this T-scope panel. And again, this is what T-scope looks like. It's big, it's also 34 colors. And again, I am sorry for having to redact some of the markers. Okay, so flex channels. Successful proof of concept of showing that your flex channel strategy works is that no matter what marker you choose, it has no impact on the rest of the panel. Does that make sense? So what we did was get 3 markers with a variety of different antigen densities, low, medium, and high. And we got them on our the two channels that we initially designated as flex channels, and these are BV421 and Percy BV471. And so you matrix these together, and you get 9 different cocktails. And to be clear, each of these 9 cocktails also includes all of the other markers in the panel too. They just differ in what's on 421 and what's on Percy BV471. So we assayed this on a variety of different material types. I'm just going to show you a single sample for clarity. But this is just a histogram showing what markers A, B, and C look like across all 9 of the cocktails that I just described. And you can see that, in general, the staining pattern is robust, and it doesn't really change depending on what your flex cocktail is. This is true for most markers in the panel, but it's not true for all of them. So when we looked at these 3 markers, these are unhelpfully labeled markers 15, 21, and 27. You can see that the blue, purple, and gray histograms show a dramatically different staining profile than the rest. And what do blue, purple, and gray have in common? These all correspond with the cocktails that have the abundant antigen on Percy BV471. So this suggests to us that this is related to the fluorophore that we chose. So in trying to understand, why this is and how can we avoid this, why do we only see this on Percy BV471 and not on 421? Those of you that are familiar with Spectral probably are not maybe surprised by this phenomenon, but we were trying to understand, what makes one marker good for flex channels and one marker bad. So we spoke with a lot of folks, and we ended up kind of cluing into this metric that Cytec estimates for you if you have a Cytec instrument. This metric is called stain index reduction. And stain index reduction is a predicted value, and what the value predicts is fittingly, according to the name, is the decrease in resolution of one marker by the spread imparted by a separate marker. And this value is calculated pairwise for each color, but what I've done here is aggregate these values for any one of the individual colors. And what you can see here is that BV421 has a very low stain index reduction, and Percy BV471 has one of the highest in the panel. So we posit that stain index reduction is a useful metric for determining what might work well as a flex channel and what might not. And indeed, when you look at all of the pairwise interactions, the text might be a little bit too small here, but essentially the point I'm trying to get across is that the intersections with the marker-marker pairs that have the highest stain index reduction happen to correspond with the worst flex issues. And so the box markers here are indeed markers 15, 21, and 27, and those with excellent memories will recall that those were the problematic histograms that I showed prior. Cool. So now we're armed with this knowledge that SIR is a useful metric to determine good and poor flex markers. So we repeated the same exercise with the all PBMC panel. The difference here is that we deliberately chose markers with low predicted stain index reduction. And so we picked BV480 and ABC. And we used more or less the same experimental setup where we matrixed 3 antigens of different densities. And what I'm showing here, and it's kind of hard to see in the bar chart. The bar chart shows the percent total for each of the populations. and they're colored by what the flex marker is. And the font is small, so it's a little hard to see. But I think an easier way to show this is the table that I'm demonstrating here, where for populations that are greater than 1% of the total, the maximum CV is less than 10% across all 9 of the cocktails, and indeed lower than 5% for most of them. So really happy with this, this interpretation. We are convinced that this setup works well for the all-PBMC panel, and we hope that it'll work well for you too, if you're interested. So in conclusion, we propose a strategy to really essentially flexibilize your existing panels. We're continuing to develop the strategy, and all of our preliminary data so far suggests that the strategy is pretty robust. The takeaways from this is that your choice of channel is really critical. Colors with high state index reduction are not recommended as flex markers. And in addition, what marker you choose in terms of its antigen abundance and proper titration will also help your flex schemas. So that about wraps it up for flex channels. I'm going to change gears again and talk about this really cool technique called, and I will speak about it again here. And before I get too far, I need to give a big shout out to Drs. Headley and Etienne Becht of the Fred Hutch Cancer Center. We've had a standing collaboration with them for some time now, helping us. They're the inventors of this technique, and they've spent a long time helping us get up and running. So Infinity Flow is really its own hour-long talk, so I will try to kind of shrink this down into a way that is digestible. So how Infinity Flow works is you take your sample, you stain it with your full stain cocktail, whatever your favorite full stain cocktail is, and you stain in bulk, and then you split your sample across all wells of these special plates called massively parallel plates. And these are available commercially from BD or BioLegend. We use the BioLegend one here, which and essentially in these massively parallel plates, each well contains a different marker, all conjugated to the same fluorophore. So this is really useful for screening, screening a whole bunch of markers at once. So within each well, we have the same sample that's stained with kind of our backbone antibodies, plus one unique infinity marker. And then we acquire our favorite flow cytometer. Following acquisition of the data, we can feed all of this data into this really nice Infinity Flow data package. And in a nutshell, what the algorithm does is predict the expression of all of the infinity markers using the scatter profiles and the backbone staining as an input. So what you get is, what you get after you run this pipeline is you get your FCS file, and it has all of your empirical collected data, but it also has 400 or so additional features that have been predicted by the Infinity Flow algorithm. So hopefully that makes sense. If there are any questions, please chat with me after the talk. So it's a great way to get super high parameter, high feature space data, and really good for investigational purposes. So one of the first ways we deployed this assay is on characterizing novel activation markers. As you might imagine, characterizing what happens to CAR Ts after they recognize their antigen is really important, and there's a lot of vested interest in figuring out, what is on the CAR T cell, either singly or in combination, that happens to the T cell after it encounters its antigen. So, and even in a simplistic analysis like I'm showing here, which what I'm showing here is the enrichment or downregulation of each of these markers upon stimulation over time. And a lot of these markers and I've subsetted to what I think are the most interesting. A lot of these are familiar to us, like PD1, LAG3, CC2L, CD69, and so on and so forth. A lot of these markers are canonical activation markers, colored in red. But there's a lot of new markers, colored in blue, that might have some utility as novel activation markers, like CD120B, OX2, CD155, CD355, all of which I'm less familiar with. So it might represent, opportunity for novel understanding of CAR T biology. So not only is Infinity Flow useful in, discovering new markers in kind of a general T-cell biology sense, but those of you in industry in the room, this is also really helpful for specific program or product investigations. So I will especially apologize here because I have to be a little vague. We observed an interesting phenotype, I can't be more specific than that, in a few samples. So the interesting phenotype is dotted in red, and the normal phenotype is under the where it says typical. So this is the phenotype that we observed is very unusual and not expected. And there's a lot of work that is trying to kind of understand what's going on with these unusual cells. We had previously ruled out technical artifacts or detection antibody-related issues or protein internalization, and the indicated cells are not dead or dying. So what are they and what's interesting about them? We can use Infinity Flow to help answer. So even simplistically, we can see what markers are enriched in these samples and what markers are down-regulated. I'm showing you that here in a simple molecular plot. But we can kind of turbocharge these data by plugging them into our more high-dimensional tools like clustering techniques. We can see which combinations of markers are maybe co-regulated, co-expressed or co-deregulated, shown in the clustering map here. And we can also plug these data into network analysis algorithms like StringDB and QIAGEN's Ingenuity pathway analysis to help add that additional kind of biological contextual layer on top of these data. So this has been really helpful in identifying and elucidating this problem. Great. So that wraps up Infinity Flow and brings me to parting remarks. Overall, I think spectral cytometry has the potential to really facilitate cell therapy characterization and offer additional insights into product quality, really important to folks in the industry. There's a whole bunch of resources that exist for you to make the most of your spectral cytometry data. I highly recommend, if you're not already paying attention to these labs or these OMIQs, to pay attention to them. Look up these labs and see what's coming out of them. There's no shortage of experts in this area. And finally, pay attention to novel techniques. Read Cytometry A, see what's kind of coming out. Infinity Flow, for example, cool new technique to take advantage of. With that, I'd like to thank you all for listening to this talk, and I'd like to thank my collaborators from BMS, Fred Hutch, and Dotmatics. And thanks for having me. Happy to answer questions, and that's all that I have. Sorry, yeah.
Sorry, apologies for the croaky voice, I've got a cold. But do you know if Infinity Flow has been validated for use in the clinical space?
That's a good question. I don't know for certain. All of the Infinity parameters are imputed, as in they're predicted. So it requires a lot of validation work that I didn't describe here to evaluate the accuracy of the prediction. There's different things you can do to your backbone panel to increase or decrease the accuracy of the prediction. The real answer to your question is I don't know. So far, to my knowledge, there haven't been that many papers that I've read that use the Infinity Flow algorithm. So the real answer is I don't know.
Okay, and just a follow-up question. The markers on the plate, are they all in the same fluorochrome?
Yes, they are. They're all conjugated to P in the case of the biology plate. Yeah.
General question for you. So you mentioned that you have your classical strategies for CAR T characterization versus the new ones with the spectral and the old ones with the conventional. Do you think in a couple years the conventional strategies will be fully phased out, or will there always be a use for them?
I think there's always going to be a use for them, especially in the context of like the QC manufacturing setting. There will always, I think, be different needs for discovery and early, attribute understanding as compared to like a more targeted assay that can maybe be run at a CRO or like a routine testing core in your organization. I think the discovery setting is going to really increase its scope in terms of spectral characterization, but I think there will always be a place for conventional cytometry. Good question.
I thought the flex flow was really cool, the flex channel idea. If you don't have cells that represent signal on every channel, you might miss something, and so then when you go to a disease sample, there's a new cell type, has really high expression, and it gives a lot of spillover into some channels. Do you have a way of controlling for that? Where it looked like you were testing on cells, do you also test on beads or something with sort of, cephalin signal levels to get that?
Yeah, that's a good question. We haven't tried it on beads. Kind of our strategy was to use like the most, in our like testing characterization that I talked about, use like the most abundant antigens that you can find, like beta-2M or CD45 or something super abundant that you don't expect to, it to possibly be brighter than that.
Thank you.
Yeah.
great talks, everybody. I'm going to express my naivete and all this stuff because I'm just getting started with it. All this wrong word already, damn. The compliance, the regulatory stuff. I saw an image with pointing to all over the world. Does everything apply all over the world? Because I'm new to this and I'm learning my P's and Q's and I's and Q's and O's and Q's.
Yeah, thanks, Peter. That's a really good question. It's you know, are the validation requirements in the U.S. the same as everywhere else, right? And you know, the answer is no, right? You know, the FDA doesn't exist everywhere. However, I think a lot of countries and a lot of places outside of the States use the FDA and our regulation standards as a model, right? So it's kind of, if it's good enough for us, it's good enough for you, sort of thing. And I think that's kind of what we see in the real world from a lot of these labs. So, all right. So if there aren't any other questions, I know we're probably out of time already. But of course, you know, thank you for coming out today. We want you to come back to